Hey, my name's Joe Luczkovich. I'm presenting this poster, basically comparing side scan imagery obtained from a recreational echo sounder with single beam sonar from that same echo sounder to use it in benthic habitat surveys for fish. And basically we're surveying submerged aquatic vegetation, or SAV, in Albemarle Sound, Pamlico Sound, and the Neuse River in North Carolina. And as you can see here, the purpose was to see if we could use side scan to get an additional view of our transect lines in seagrass and SAV areas along the coast of these three estuaries. So we have been using a single beam, which is a downward looking sonar beam. And you see in this image right here, what you would see on the screen of your echo sounder. It tells you, your lat and long from a GPS. It tells you the heading and your speed. We go about 3 knots, between 2 or 3 knots. And it tells you the depth in meters. And then you see on the echogram a grass bed. In areas where there's no grass, you would just see a flat sort of red hard line, which echoes off the bottom if it's sand or mud. But where there's grass, there's a jagged kind of canopy that's visible. And so in areas where there's a lot of SAV, they're shown in red areas in this upper chart. Oh, this is in Kitty Hawk Bay, where we have a big grass bed right near the Outer Banks of North Carolina. And the redder this bar is, the redder the color ramp, the more SAV bio volume there is in a sentinel site or in a longshore transect. So here's another rapid assessment transect, which we did along the entire coastline of those 3 estuaries to find out where the SAV is densest. And that single beam sonar shows it in a 25 meter wide band, but actually the sonar is actually just looking straight down below the boat. Gets an accurate depth, and it tells you whether there's SAV under the vessel, but it doesn't talk about whether SAV occurs to the side of the vessel. So this is a little bit misleading. However, the side scan taken along that same track does show variations when you look at it a little more closely. Like this is a down scan, and this is a single beam down scan, and this is a side scan view of the same location right under the boat. That's what it would look like if looking down. But off to the side, you can see there's big patches of seagrass. And we've done whole sentinel sites back and forth, and then characterized the presence of areas like this using a hand-drawn polygon in Google Earth. So just in summary, there's good agreement between the down viewing single beam sonar and the side viewing side scan sonar. The side scan sonar goes out to 12 to 14 meters on either side of the boat. So it is reflective of what's happening inside that scan of 25 meter wide swath. Now, this is just some statistical analysis to demonstrate that. You should look at the poster carefully, but we had 68 agreements between side scan and single scan on the SAV positive areas. And when SAV is not present, they agreed 38 times out of, I think this is 120. So there were a few disagreements. That's to be expected. But in general, the side scan gives a more conservative estimate than the interpolated single beam sonar at our sentinel sites. So we have been using the single beam. We may be overestimating grass beds, but actually I think it's better to be a little bit conservative on the overestimating side. But if you wanted to use side scan to give you more confidence, we think it's doable. And if you have questions, please contact or chat with us in the chat box. Joe Luczkovich and Noah Gwynn, Department of Biology, East Carolina University. Thanks. Okay, I want to show you some pictures of our sonar. This is the Lowrance and Total Scan. This is the DTX from Biosonics. We run 40 shore normal transects at a sentinel site. This is the Trent River site, and you get a map from Biobase that shows where the SAV is densest. That's the red areas on the map. And then you can do change over time. That's Edenton, North Carolina, Albemarle Sound, a 5-year period. We lost all the SAV. You get a longshore map of SAV density as well. And this is Kitty Hawk Bay. And then with Reef Master, we use the same transect to look at side scan sonar along those longshore tracks. Now some field work.
Hey, where are you guys?
Albemarle Sound.
What's the name of your team?
The SAV squared.
What does that mean?
It's submerged aquatic vegetation. And then it's also, the second part's the
Sonar and video.
Team.
Video
Yeah,
team.
sonar and video team. All right. And yeah, I'm here with them. This is Dr. Joe Luczkovich, East Carolina University. Our vessel is the RV Pinfish. And we're at Albemarle Sound at Sandy Point. Say goodbye. Say, go Pirates.
Go Pirates! Arr!
Dan Crane and Hildy Spate, Tyler Peacock, Allie Mulligan, John Sherman, Noah Gwynn,
Instagrammer.
Setting up the boom that we use for our transducers on our new rig. This is a coring at one of our Sentinel sites. We do sonar first. We have one person on the boat. We have 2 others here, myself and Noah Gwynn, in the water, doing quadrat surveys and taking core samples for species composition. This is at one of the Neuse River Sentinel sites, the Cahookee Creek area. In this location, we were finding Xanakelia. And some places it was very dense, and other places quite sparse. What we're showing here is I take that 6-inch PVC core, and then we sieve it. We don't save any of the marine animals. We're focused on the SAV plants only in this, and we put it in a baggie, labeled, and with a little water from the field site. And then we take it back to the lab.
Cecilia is hooking up the sonar equipment. This is a Biosonics unit, DTX echo sounder, and there are 2 transducers, a 400 and a 200 kilohertz transducer on that biofin. A cable gets attached to each one, and just like that, it gets attached. Okay, there's seagrass there or there's not. Actually, it all gets stored on a laptop, and you run the laptop software called Echo SAV to do an analysis almost immediately after getting off the boat. Within 24 hours, you can have a map of the seagrasses.
So once that whole sonar rig is set up, you got the laptop running, you put the transducers on the fin in the water, and you run your transect. Last thing we do is go to 100 video verification points with a drop camera. Shown here, we visit these. These are randomly selected points in our map area, and if the bottom has no SAV, it's pretty clear. Even though the water itself is kind of turbid, so that location is sand bottom, so that's no SAV. But then other locations have SAV, and we record that as present. We also record the depth and the lat and long. We now have a system that has a built-in GPS. So we get a map of true positives, true negatives, false positives, and false negatives. I'd like to thank East Carolina University, the Albemarle-Pamlico National Estuarine Partnership, North Carolina Division of Marine Fisheries, the NOAA, and the National Fish and Wildlife Foundation, and North Carolina Sea Grant. All of those agencies help provide funding and support for this project. Thanks for your time. Bye.